Well, hello, everybody, and welcome to today's webinar. I'm Abby from Precisionary Instruments, and I'm really glad you could join us. It's my pleasure today to introduce our speaker, Dr. Brenton Laing, who is an assistant professor of pharmacology at the University of Mississippi. And we were just talking about how wonderful living down the South is. Dr. Laing leads a research program focused on the brain circuits that regulate survival behaviors, especially how these circuits can become dysregulated under stress or substance misuse. His work explores the anterior hypothalamic area, and he has some exciting findings on fast spiking parvalbumin neurons and their role in behavioral responses to threat. So today he'll be sharing his research on the causes and consequences of anterior hypothalamic area activity. And we're really looking forward to hearing more about how his electrophysiological studies of these neurons are linked to behavior during and after threat exposure. And really quick, Dr. Laing earned his PhD in bioenergetics from East Carolina University. He completed his postdoc training at the National Institute on Drug Abuse with Dr. Yeka Aponte, where he investigated neurocomputational systems related to threat and reward processing. And with that, I'd like to turn it over to you, Professor Laing, and thank you again for your time today.
That's a very kind introduction. Thank you for that, Abby, and welcome to the guests who are attending the seminar today. I titled this one, Controlling Consequences of Anterior Hypothalamic Area Mediated Escape. So if I can't see the chat box during the talk, but if at any time anyone wants to shout out a question or a comment, feel free to chime in. Otherwise, we'll get the comments at the end. My lab is really focused on how the hypothalamus controls highly conserved survival behaviors, such as fight or flight behaviors, as well as feeding behaviors. And we're trying to dissect the hypothalamic circuitry that controls these survival behaviors. So I love nature. I love always looking to nature for things that can drive my research questions. And I always like to start this talk with this example from a news article where this rodent is approaching this snake, doing this investigation, and clearly pretty curious, getting its nose all the way up to this snake before realizing that, oh no, there is a threatening situation here. This snake may try to eat me. So at that moment, the rodent kind of jumps and tries to get out of the potential reach of the snake. And I'm really interested in what kinds of brain circuits control this moment of threat detection and the escape behavior that follows, as well as the consequences of the activation of this circuitry along longer time scales. All right, so I really became interested in this area as an undergraduate with the seminal work by Dr. Sapolsky, Why Zebras Don't Get Ulcers, that was curious about how is it that a zebra can be feeding on grass in the Serengeti when a lion jumps out, do this escape behavior, but then as soon as they escape safely, go right back to grazing. To me, that's a remarkable cycle that, as he states, is oftentimes leads to problematic consequences or pathological consequences in humans because of the degree to which we ruminate on our past exposure to threats. Once we start to ruminate for extended periods of time, these fight or flight responses, which are evolutionarily designed to be transient or short-term in nature, become prolonged and start to have harmful effects that are distributed across many organ systems and can result in damage to, cardiac function, liver function, renal function, for example, as outlined by the late great Bruce McEwen. So, as I said, I'm really interested in the hypothalamic area, the hypothalamus, and for a little bit of background, the hypothalamus is comprised of many subnuclei, each of which is thought to be responsible for different aspects of survival. There's this subregion known as the anterior hypothalamic area, which is thought to be involved in thermoregulation and defensive behavior. But really, very little is known about this anterior hypothalamic area. What are the synaptic inputs that control it, and what are the molecularly defined populations within this region that can modulate physiology and behavior? So we set out to explore the role of this AHA, here described as AHN, in survival behaviors, and stumbled upon these profound defensive behaviors in response to threat. So some very seminal work by Dr. David Anderson found that this region nearby the anterior hypothalamus, known as the ventromedial hypothalamus, has a population of neurons that become activated during a threat. So here he's doing a fibrophotometry experiment where the mice are exposed to a rat or a toy. Rats are predators for mice, and so you get this big activation of the ventromedial hypothalamic area or ventromedial hypothalamus during this rat exposure, quantified here. What was really interesting about this finding is that he continued to monitor the anxiety behaviors of the animal after the rat and the relationship that these exposures have in driving those behaviors. And what he found is that after the rat, there's a change in the anxiety behaviors of these mice, which were exposed, that persists long after the change in the activity of the VMH. So there's this long-term state change that underlies what he calls a persistent defensive state or a persistent emotional state of these mice, which can drive network level effects throughout the brain. Interestingly, some other work by Dayu Lin's group showed that this ventromedial hypothalamic area sends a monosynaptic projection to the anterior hypothalamic area, as well as this periaqueductal gray, and that the VMH to PAG projection controls freezing behavior, and the VMH to AHA projection controls escape behavior. So we've got this innate threat detection system that sends projections to this anterior hypothalamic area. And then another set of work by Bang showed that the hippocampus can project to the anterior hypothalamic area and convey stored long-term memories. And so in this way, there's information about learned processes that are feeding into the anterior hypothalamic area and information about innate processes that are feeding into the anterior hypothalamic area. So we set out to understand how do the activity patterns of the AHA change during exposure to some kind of threat. So first, we use this classic fear conditioning paradigm while we're doing miniscope imaging of the anterior hypothalamic area. So here you can see GCAMP7 expressed in the AHA and the track of the grin lens, which was implanted into this mouse's brain. And this technology allows us to monitor the activity pattern of these neurons as the mice are freely moving in the environment, investigating, exploring, and also being exposed to stimuli. So we set up this shock paired conditioning paradigm where on day one, mice are exposed to this neutral tone followed by this shock. There's a brief break and then another neutral tone followed by this shock, which is going to cause an association to form for the shock onto the formerly neutral tone, now conditioned stimulus of the tone. And then on day two, we put the mice back into the apparatus and we present that tone again. And we ask the question, what are the changes in activity patterns that we can observe during tone delivery on day two? So here you can see this heat map of many different neurons. Each row is an individual neuron. Here are the two trials of the shock exposure. And you can see this dotted line is indicating the start of the shock or onset of the shock. And you get this activation pattern of the AHA, which is greater on day one than greater than day 2, as I quantify in this manuscript. But then what we see on day 2 is that there is an activation of the cells during the tone delivery that we didn't see on day 1. And so here's quantification of each of these, where we have a pre-tone phase followed by a shock delivery, which causes a very large rise in activity, and then a second shock on day 1 that causes a significant but smaller shock than the first shock. And we found that the change in activity pattern of these neurons is highly correlated with the speed of the animal, the locomotor speed of the animal. And you can visualize on this graph where the shock phase is. The mice are moving very rapidly, where the post-shock phase is, and where the pre-shock phase is, which is a pretty striking relationship. So here we use this classic measure of time spent freezing, which indicates that our mice successfully acquired the conditioned fear during tone exposure. So our paradigm worked, and then we could ask the question, what happens to the activity pattern following the tone on day 2? And so here is the tracer with the mean, and these are error envelopes across all the cells. And what we see is that there is a significantly higher or significant increase in activity following tone delivery on day 2 compared to day 1, quantified here across the cells. In addition, we can quantify this pre-area to this post-area on day 2, and we see significant elevation. So taken together, this experiment showed us that the AHA becomes activated during the presentation of a learned fear stimulus. In addition, we looked at the behaviors of the mice during this session, and one of them that stuck out to us was the grooming behavior. So here is a peri-event stimulus plot where we basically defined the moment of grooming onset as time point 0, and we are visualizing the activity of the cells here on the y-axis. And so across all of the bouts of grooming, if you average across the mice, then you get a significant reduction in activity following the onset of grooming compared to the moment that precedes the grooming. So what we see here is following the activation of the AHA by this conditioned threat, when they transition to a grooming behavior, there's a decrease in behavior, creating this kind of peak and trough effect that seems to be related to either locomotion or grooming behavior. So we further analyzed this data using SEBRA for joint analysis of the neural data and grooming data. So basically, we did this with all 8 mice. This just shows representative 4 of them. But here's the observed data. So each of these embeddings is a model that's trained on one mouse and tested on a totally different mouse. And then here are the shuffled embeddings where we basically have fully randomized the assortment of activity patterns and grooming behavior. And the black dots are the samples where there is no grooming behavior, and the red dots are the samples where there is grooming behavior. What you can see is that across these mice, there is a much smaller cluster of red dots in the observed data sets than in the shuffled data sets, where they're basically randomly distributed. And so we can calculate this with a number of different measures. The kind of gold standard for this is the Euclidean distance, which basically takes the sum of all of the points within the embedding that are red dots. And you can see that the cluster is much smaller, quantified here. So what this data means is that there's a representation of grooming behavior in the activity pattern of the AHA, which makes it likely that there's some contribution to the generation of that behavior from the AHA. In addition, if we split our mice into mice that freeze or do not freeze during the tone presentation, we actually can see different activity patterns, such that the mice which freeze have this gradual rise in AHA, whereas the mice that do not freeze have this huge drop followed by this big activation. And when we train machine models to be able to classify if a mouse is a freezer or a non-freezer based on these activity patterns, we're able to do this well above chance. So there's information in the AHA related to the freezing decision that a mouse makes. So then we sought to directly manipulate the anterior hypothalamic area activity patterns. And we use this strategy of optogenetic activation, whereby we put channel rhodopsin into that presynaptic ventromedial hypothalamus and implanted fiber optics over this postsynaptic region where you've got dense axon terminals. And there's a schematic here. So we had two groups. Basically, one group has a functional opsin with a fluorophore that's green that you see here, and another group that only expresses the fluorophore. And we're going to run some behavior experiments in these mice to figure out what does activation of this circuit that is excitatory for the AHA have on behavior. All right, so here's some example videos. Actually, one of the first tests we like to run is this open field test, where we basically just attach this mouse to this patch cord, which is where the laser stimulation is delivered through that fiber optic to the AHA, and allow the mouse to freely explore an open apparatus. Now, a typical mouse will just kind of wander around the apparatus, maybe digging, smelling, looking around. But what we saw was a very robust jumping behavior in these mice, very similar to what Dr. Lin had shown from this projection. So at first, then we asked ourselves, okay, is this just a motor response where all of a sudden the mouse is just jumping around uncontrollably, or is this some kind of goal-directed response to try and either escape through the clear wall that they maybe can't perceive or get over it, or, you know, what exactly are we looking at here in this jumping behavior? So we set up this real-time place preference apparatus, which is kind of dojo style with a line down the middle. And basically, when the mouse crosses over to this side, the laser turns on. And when the mouse crosses over to this side, the laser turns off. So in this way, the mouse can choose if it wants to avoid the laser stimulation. And what we observe is the mouse rapidly returns to the non-laser paired side of the apparatus. So test it again. And so this assay was striking for a few reasons. The first big reason is that we didn't observe the jumping behavior. We got a totally different selection from the repertoire, that the mouse is just turning around and walking away, which was our first clue that this is not a jumping circuit per se. This is a circuit of avoidance. And if there's opportunity to avoid the stimulation that is easy, the mice will take it. However, we wanted to test this circuit in a more ethologically relevant environment. So we used this elevated plus maze. This is a classic test for anxiety, whereby you've got two closed arms on the horizontal arm here and two open arms on the vertical arm. And mice tend to prefer closed spaces. So a mouse that's anxious will be more likely to spend time in this closed space compared to a mouse that is on an anxiolytic, like a benzodiazepine, for example, or has a high motivational drive, will be more likely to walk out to these ledges and peer over the side. So we deliver stimulation to these mice. And the mouse evacuates the elevated plus maze. So I've only seen one report in the literature where someone suggested that this happens, and it was like mice that were literal mice that they found from the wild and brought into the apparatuses. So this is very unusual behavior for a C57 BL6 mouse. And a little bit counterintuitive, because over here we're saying, okay, it's driving avoidance behavior. It doesn't look like it's enjoying the stimulation. So perhaps we expected that the mice would sit in this closed arm because they're very anxious. But instead, what we saw is the vast majority of the mice just evacuate off of the maze. And actually, so many evacuate that it's hard to even quantify the the elevated plus maze data because none of them even make it to the end of the test, whereas all of the controls make it to the end of the test. Okay, so we did some quantification of the behaviors during this the real-time place preference assay. And so here are some heat maps. And you can see for the YFP mice, they prefer the corners, but they're approximately equal between the YFP or the laser on and the laser off side. Whereas the channel rhodopsin mice really prefer this laser off side of the apparatus, quantified here. The average duration of visit to the on zone was much shorter in the channel rhodopsin. They crossed the line and then they return right back to the laser off side. One thing that was really interesting to us is we didn't see a significant decrease in the entries to on zone. So it seems that the mice are still willing to investigate whether or not this side is worthwhile to test in a way that is very different from a shock paradigm where the mice would just learn to completely avoid that side of the apparatus. In addition, one thing that we noticed is that there is a significant increase in time immobile of these mice that have the stimulation. So we further analyzed, well, what are they doing when they're immobile? So immobile means that they're not moving in XY space of the apparatus, but they're not frozen. They can be physically either peering around or grooming themselves, for example. So what we found is that there's a significant increase in grooming behavior of the mice following this stimulation on the laser off side. So this was really interesting because it kind of matched our calcium imaging data where you've got this activation followed by this subsequent drop in activity when the laser shuts off, which results in the onset of grooming behavior. And this grooming behavior is said to be a displacement behavior in that it displaces the time they would spend doing other things, such as digging and rearing. They're spending less time investigating their environment and more time focused internally or focused on themselves. So then we set out to understand what happens if we do this stimulation for longer periods of time. In a very short test, 20 minutes, real-time place preference, okay, maybe the consequences are fairly short-lived, as in the case of a zebra on the Serengeti. But what happens if we repeatedly administer this sorry about that, my microphone dropped. If we repeatedly administer this stimulation to mice over long durations, so for 3 days straight, we administered 3 hours of stimulation to the mice, and then we continued to track them via video, but we shut off the laser. So the stimulation paradigm was this fixed ratio, or sorry, fixed interval, 5 seconds on, 25 seconds off. So they're getting 10 seconds of stimulation every minute for 3 hours. And then we shut off the laser and we continue observing the behavior of the mice. And what we see is that there's this significant increase in grooming behavior post-session, even compared to the grooming behavior during the session. And both of those are significantly elevated compared to their fluorophore counterparts. So what we can see is that at least the behavioral changes being driven by this activity pattern of optogenetic stimulation are persistent, much like what David Anderson saw in the VMH when mice were exposed to the rat threat. So all of those experiments were in awake mice while they're freely moving around apparatus. But we wanted to better understand what were the networks of circuits that were attached to this stimulation. So we set out to do this PET experiment using fluorinated deoxyglucose. So basically, we anesthetize the mice so they should have no concept that they're receiving the stimulation. They go into this brain scanner, and then we inject them with this FDG that should get uptaken into areas of the brain which are active, shown as hotspots with this radioactive form of glucose. And what we saw is a significant elevation of activity in the amygdala as well as the striatum. So even in fully anesthetized mice, we're getting the downstream activation of nodes responsible for things like fear, anxiety, as well as repetitive behaviors. So we found this to be a really exciting thing, that anesthetized mice were still getting the same network level effects. It's not like there's some perception by the mouse that there's a thing in the environment that's giving me stimulation, and then I should be afraid of that. These are potential domino effects within the brain. So as a summary, VMH to AHA stimulation promotes jumping behavior, locomotion, and avoidance. It triggers repetitive grooming that follows the stimulation and persists. And the VMH stimulation increases activation of the amygdala and ventral striatum. So once we knew that the anterior hypothalamic area was involved in these threat behaviors, we set out to understand what kinds of cells were in the AHA. And one that really stood out to us was this population of parvalbumin expressing neurons in the AHA. There's only around 400 of them, which is a relatively small proportion of the total number of cells in the AHA. But parvalbumin neurons elsewhere in the brain can be highly connected regulators of postsynaptic targets. So we set out to understand what is the identity of these cells, what do they connect to, and what are the effects of activating them. So first, we did these radioactive in situ hybridizations for markers of VGLU2 and VGAT. And what we found surprised us, that there was a high degree of co-localization with the VGLU2 marker, but almost no co-localization with the VGAT marker, quantified here. So at least at the transcript level, these cells appeared to be glutamatergic neurons, not GABAergic neurons, even though elsewhere in the brain, almost everywhere, parvalbumin neurons are GABAergic. So because this was so surprising, we sought to use a second method. Basically, I took VGLU2 Cre mice or VGAT Cre mice and injected them with a Cre-dependent virus to express green fluorescent protein, and then did an immunostain for parvalbumin. And what we found was a very similar result to our in situ data, that we got co-localization of the parvalbumin with the glutamatergic marker, but almost no co-localization with the GABAergic marker, even though there's many more GABAergic cells within the AHA. So we said, okay, this is pretty exciting. We've got a glutamatergic population of around 400 parvalbumin neurons. What do they do? Where do they connect? So the first thing we did is another mini scope experiment where this mouse is connected to this miniature microscope, and we ran a 3-stage test where we took a baseline and then we drop in this rat. And so then there's this threat exposure, and then we remove this rat. Again, these mice are expressing GCAMP for us to be able to monitor the neuronal activity. And we did this in both the VGAT and the parvalbumin mouse lines, VGAT Cre and parvalbumin mouse lines, so that we could look at changes in activity patterns in both of the populations I just showed you. So here in the top panel is the results from the VGAT. We didn't see any significant effects of threat exposure on the VGAT mice, whereas for the parvalbumin mice, we saw a significant increase during threat exposure that decreased during threat after the threat was removed during the assessment of the wall space. So taken together, we saw by parametric analysis, at least, the parvalbumin neuron activity changes during rat threat exposure. In addition, we did some machine modeling to try and predict whether or not the rat was in the apparatus based only on the activity patterns. And we did this for both the PV neurons as well as the VGAT neurons. So here you can see a confusion matrix with the baseline, threat exposure, and assessment stages of the prediction. And here are the true classes. And you get this nice orthogonal, which indicates that these parvalbumin neurons are pretty good at being able to sort which stage of the test the mouse was in. Particularly effective for this threat exposure stage, to be able to tell if the rat is in the apparatus or not. Interestingly, the VGAT cells were also able to make this kind of classification well above chance. And so It seems that at least the VGAT neurons may hold some threat-related information. Perhaps it's a heterogeneous population where some are increasing and some are decreasing that is kind of clouding your ability to detect an effect here. So we think that this is a potentially interesting thing to follow up on to dissect the populations within these VGAT cells. But regardless, we were really excited about this parvalbumin effect and set out to understand more about these cells in particular. So the first thing we did is another one of these PET experiments where I injected channel rhodopsin or a fluorophore control into the AHA of parvalbumin cream mice, implanted fiber optic, anesthetized them, deliver stimulation, and then we look throughout the brain to find changes in activity patterns. And what we found is an increase in amygdala activation following this stimulation in the channel rhodopsin mice compared to the controls. So very similar to what we saw in the VMH to AHA projection, the 400 parvalbumin neurons, or some population of parvalbumin neurons, is able to evoke somewhat comparable amygdala activation effects. So we found that to be a really exciting parallel between our studies. I did an electrophysiological characterization of these parvalbumin neurons. One thing that's very interesting about parvalbumin neurons elsewhere in the brain is they have this fast spiking phenotype that can fire upwards of 100 hertz in many locations. So I did current clamp and voltage clamp recordings of these neurons, did a whole characterization of many different action potential and intrinsic membrane properties, and confirmed indeed that they have this fast spiking phenotype observed elsewhere in the brain. I followed this up with an in situ hybridization using RNA-scope for parvalbumin, as well as Kv3.1, which is a potassium channel involved in the rapid depolarization following an action potential, as well as HCN2, which is a hyperpolarization gated channel that is responsible for this IH or SAG that you can observe here, which is this rapid depolarization following a hyperpolarization event. So these parvalbumin neurons have the molecular characteristics that we see in parvalbumin neurons elsewhere. They have functional properties that are similar to what we see elsewhere, but they're glutamatergic, which makes them quite unique. So then we set out to understand where do they connect to in the brain. And around this time, I had a conversation with one of my colleagues at Opto GRC from Dr. Adhikari's lab, and he explained to me this experiment that they're doing where they exposed mice to this rat. And there's this ladder and a burrow, so the mouse can go investigate this rat. Complex escape behavior. So this is a really important study that showed the premammillary dorsal nucleus was involved in escape behavior during threat exposure. We did anterograde tracing of the AHA parvalbumin neurons. So I put this synaptophyte infused M-cherry into the AHA of parvalbumin cream mice. And then we look all throughout the brain to figure out where are the synaptic projections of these neurons. And we saw dense axonal projections in the AHA, or in the PMD, excuse me, premammillary dorsal nucleus. So that region that Avishek's group very elegantly showed us in control of threat-driven escape behavior is receiving a direct excitatory projection from these AHA parvalbumin neurons. We further verified this by putting a retrograde tracer into the PMD of a parvalbumin reporter mouse and showed that there's around 50 to 75% of those parvalbumin neurons that are backfilled, indicating that they project to that PMD region. So we kind of forwards and backwards verified the axonal circuitry is in place, at least, for this to be a functional projection. Then we conducted Crackham experiments, or channel rhodopsin assisted circuit mapping using slice electrophysiology. I put channel rhodopsin into these AHA-PV neurons, as I showed you in the PET experiment. And then I patched to cells in the PMD and briefly shined this blue light to depolarize these synaptic terminals and visualize the postsynaptic effects. what we observed was an excitatory postsynaptic current that could be blocked by AMPA receptor CNQX in these PMD neurons. This both confirmed our anterograde tracing data as well as our in situ hybridization and transgenic strategies that these AHAPV neurons are excitatory because this is a glutamatergic receptor. This is a glutamate-dependent effect. So this is in a brain slice. We wanted to ask this question, could the AHAPV neurons activate PMD cells in vivo? So I stimulated mice for 1 hour before euthanizing them in this channel-rhodopsin group compared to controls, which is just the YFP. You can see these axon fields in both groups. And then we do this immunostaining for immediate early gene FOS and compare between groups. And what we got is a significant increase in FOS following the 1 hour of photostimulation of those synaptic terminals in the channel-rhodopsin group compared to the YFP group, indicating that in vivo, the AHAPV neurons can activate the PMD. So knowing that this circuit is excitatory, postsynaptic circuit controls escape behavior, we set out to formally test this. So we used a 4-group design with an inhibitory opsin IC++, a control group, which is fluorophore, and then channel rhodopsin with a fiber optic implanted over the AHA, or channel rhodopsin with a fiber optic implanted over the PMD. And you can see these maps that are showing individual segmented viral injection locations that are stacked on top of each other. So a darker image indicates more injections localized to that region. So the first thing that we did was we ran this real-time place preference, and the really only significant effect we found was an avoidance behavior of the channel rhodopsin soma stimulation mice, where they preferred the off zone compared to the on zone, as we observed in the VMH to AHA experiment. And we saw a significantly elevated time immobile in the off zone compared to the on zone in the same assay. We also saw a significant increase in speed of the mice when they did cross on to the laser on side. They locomoted significantly faster than the off side. But taken together, we basically saw the avoidance behavior from this AHAPV population in this RTPP assay, as we saw in the VMH to AHA experiment. But notably, this is a smaller magnitude effect than we saw in the VMH to AHA, where that was like a 90% avoidance, and this is more like a 65-35 avoidance. So we decided to do this more ethologically relevant experiment using this simulated predator looming task, where mice have this safety net that we acclimate them to, and they make a claim that this is their safety nest. And then we have a monitor over top of this apparatus after 3 days of habituation. On the 4th day, and after sitting in the apparatus for a while, they eventually investigate this corner. And at the moment that the mouse walks out to this corner, we start this stimulus, which is an expanding black disc that's that provides the appearance of some kind of predator, like a hawk, flying in on them, that causes them to locomote into the safety nest and escape to safety. So this highly controlled simulated assay was something we used to be able to test our circuit. And what we found is in this peri-event plot shown, at 5 seconds is when the looming stimulus was delivered. And here on the y-axis, you can see distance to safety nest. So in both the soma stimulation and the PMD stimulation group, there was a more rapid return to safety nest compared to both the control and the IC++ groups, which is quantified here across the groups using area under curve. And we generated these raster plots of when the locomotion, the escape behavior actually occurred. So we see that this AHAPV soma stimulation or PMD activation can drive escape behavior to an ethologically relevant predator. Then we use this strategy to neuronally ablate those parvalbumin neurons using this TACASP3 virus that deletes the neurons in a Cre-dependent fashion, inject this virus or a control to parvalbumin Cre mice and the AHA on both sides. And we got a significant ablation of these PV neurons. Admittedly, it was kind of distributed from this very low one to approximately a half deletion. And what we saw is a right shift during this predator looming assay, whereby the mice that were missing the parvalbumin neurons were less effective in returning to their safety nest than the mice that had fully intact parvalbumin neurons, quantified here. So the story for summary 2 is that during some kind of predatory threat exposure, these AHA-PV neurons become active, cause them to release glutamate that activates this PMD, and then this PMD can drive escape behavior in these mice to help them survive predatory threats. So overall, we've seen that the AHA becomes activated during learn and predatory threat exposure, and that activation promotes flight behaviors. Following the rise in activity, there is a onset of grooming behavior that coincides with the drop in activity, and the PV neurons in the AHA contribute to threat detection via their projections to the PMD. With that, I'd like to just acknowledge my team. I have 2 PhD students. We're working on numerous software with a team of computer scientists that work in my lab, developing some wireless technology and running some additional behavior experiments. With that, I'll take any questions.
Hi, thank you so much, Dr. Laing. I don't see any comments or questions in our guest webinar attendance, but I had one. In the very beginning, you mentioned Robert Sapolsky's studies motivating you. I actually had him in undergrad and took his class and read his book, Why Zebras Don't Get Ulcers. And he has studies were on stress and hormones. I was wondering, given your work in the AHA and parvalbumin neurons, how do you think circulating stress hormones like cortisol modulate the activity of these neurons or influence the behavioral outcomes you observed in your threat paradigms? And I know this might be like way out of the scope of it. It's just nice. I just wanted to get your take on it.
No, I appreciate that question very much, and it's completely within scope. So my group is very actively working on these experiments, and we're not the data's not published yet, so we're not prepared to fully release it. But
Okay.
the way I think about this is on actually a different, the opposite direction. It's a different time scale. So the neurons are rapidly responding to the threat in the environment, and I'm really interested in what is the effect of the activation on those circulating factor concentrations. So rather than what is the court doing to the AHA-PV neurons, one could ask the question, what are the AHA-PV neurons doing to the court, or maybe inflammatory cytokines or other markers that then predispose you to diseases? And this is a really hot area that we're actively working in.
Cool.
Thanks for the question.
Very wonderful. Well, with that, I'd like to wrap up and just say a huge thank you for your time and for sharing your studies. And some of it sounds like is in the works, so I'll keep an eye out, and the whole company will as well.
Thanks, guys.
Thank you so much.